These PowerPoint slides talk about the meninges, which are the connective tissue coverings over the central nervous system, as well as cerebrospinal fluid, which we abbreviate CSF. The meninges are the connective tissue coverings of the central nervous system. They are found covering both the brain and the spinal cord, and form three layers. The most external layer is the dura mater, which means tough mother. The middle layer is the arachnoid. It's named for the fact that it is thin and cobwebby, sort of like a spider web. And the innermost, thinnest layer is the pia mater, which means the delicate mother. So those three layers are from outside to inside. This slide lists the three layers of the meninges, dura mater, arachnoid, and pia mater, as well as the spaces that are found external and internal to these three layers of meninges. External to the dura mater, so between the skull and the dura mater, is the epidural space. This contains nerve roots, and epidural anesthesia is put into this space. Between the dura and the arachnoid is a space called the subdural space. This space is basically empty. And between the arachnoid and the pia mater is a space called the subarachnoid space. This space contains the cerebrospinal fluid. The dura mater is thick and tough like canvas. At some places, it can be separated into two sublayers, the periosteal dura, which lines the inside of the skull, and the meningeal dura, which is more internal to the periosteal dura. So periosteal and meningeal dura are two sublayers of the dura mater. Periosteal dura is external and meningeal dura is more internal. These two layers are fused almost everywhere except at some places where they are separated, forming spaces called dural sinuses. These sinuses contain cerebrospinal fluid and as well as venous blood. The cerebrospinal fluid that is found in the dural sinuses has drained into the dural sinuses from the subarachnoid space. So this is the way that used cerebrospinal fluid is returned to the venous system, by way of the dural sinuses, which ultimately drain back to the venous system. There are three major dural sinuses which we will identify in lab. They are the superior sagittal sinus, the inferior sagittal sinus, and the transverse sinus. All of these drain venous blood from the brain back to the venous system, so they act like veins, although technically they are not considered veins because of their anatomical structure. They are formed by a gap between the two layers of dura mater. They also contain used cerebrospinal fluid that is draining from the subarachnoid space into the dural sinuses and is therefore returned to the venous system. Cerebrospinal fluid is a watery fluid. You have about 100 milliliters or so of it in each person. It is produced within the ventricles inside the brain by the ependymal cells, which are one of the glial cells that we learned earlier. Cerebrospinal fluid circulates through the ventricles and in the subarachnoid space around the brain and spinal cord, and it circulates because of movement of the body. It provides a watery protective cushion for the central nervous system. This slide shows the brain's ventricles. There are two lateral ventricles, one in each cerebral hemisphere, so a right and a left lateral ventricle. There is one third ventricle located in the center of the brain, and there is one fourth ventricle located between the pons and the cerebellum. This picture shows an anterior view of the brain, and you can see here the right and left lateral ventricles, one in each cerebral hemisphere, the third ventricle in the center, and the fourth ventricle right in front of the cerebellum. The third and fourth ventricles are connected by a tube called the cerebral aqueduct. This slide summarizes the flow of cerebrospinal fluid which is made in all four ventricles. The drainage pattern is shown by the numbers on this slide. So from the lateral ventricle, cerebrospinal fluid flows to the third ventricle, from there through the cerebral aqueduct to the fourth ventricle. From the fourth ventricle, small holes allow the cerebrospinal fluid to exit the fourth ventricle and enter the subarachnoid space, from where it circulates all around the brain and spinal cord. The used cerebrospinal fluid then drains from the subarachnoid space into the dural sinuses, which you can see at the top of the screen where number 8 is, and therefore it is returned to the blood.